This is the last seminar, sort of obviously, for this year. We will be breaking, of course, over winter break, and I believe we start up again on January 14th. And then we'll go through the end of April for next semester. So there is sort of a pseudo sign-in sheet. We'll pass that around. The official ones are sort of AWOL right now with the construction going on. So if you could sign in, though, that would be great. And with that, I'd like to introduce Wallace Chan, who's going to talk to us about ligand-based virtual screen for G-protein coupled receptors.
Okay, thanks for the introduction, Marcy. So my name is Wallace Chan, as Marcy said here. I'm with a PhD student in the Department of Biological Chemistry, and my mentor is Professor Yang Zhang. So I'm going to start the talk off here with a little introduction to GPCRs and everything related to GPCRs. So G-protein coupled receptors, otherwise GPCRs, which I'll call them from this point onwards, are a really large superfamily of receptors in the human proteome. They're responsible for many really important physiological processes, such as, obviously vision. You have your fight or flight response. You have analgesia. So, they're a very large family of proteins, and not surprisingly, they're also very often commonly associated with many sorts of diseases, such as cancer and diabetes, either through maybe mutations in the primary structure, or maybe an overactive promoter and whatnot. So not surprisingly, these are really common and popular drug targets. So let's put this a little bit in perspective here. So the human proteome has roughly 20,000 plus or minus or so proteins, right? So out of these here, in humans, this is in humans, and so out of these, roughly 825 to between 1,000, it really depends on where you get your source, are GPCRs. And out of these 825 GPCRs, roughly 113 of these are actually drugged or druggable, meaning there have been drugs released for these GPCRs. And furthermore, roughly 30% of GPCRs or 30% of drugs on the market today target GPCRs. This number really varies between 30 to 50% depending on the source that you read from, but this is generally the estimate. So therefore, it's a very attractive target for drug development. Now, in terms of development, drug design, what have you, still is today is the high-throughput screen. And what is high-throughput screening? Essentially, you have a target protein, in this case, like a receptor usually, and you screen a really large library of compounds, chemicals, against it to see whether or not there will be an effect. So this can be thousands to millions, depending on what kind of campaign that you have. But this can be very expensive process. It can be very time-consuming and also very laborious. So there have been methods developed recently that have tried to address these drawbacks of this method here as a complementary parallel approach, and which is known as computer-aided drug design, CAD. And this method essentially aims to predict whether a candidate compound will interact with your receptor structure or just target in general in any way. And usually uses a combined approach, like a bioinformatics / chemoinformatics approach, right? And so in general, we refer to these approaches as virtual screens. And virtual screens can have there are two main sort of ways in which you can approach the methodology. And you have what's called structure-based screens, and you have ligand-based screens. And I'm going to go into each of these and just talk a little bit about what these are. So first, we have a structure-based virtual screen. And what this is, essentially, is it uses information from either the structure of a known protein, that you know the structure from PDB, or if you don't know the structure, you might even go so as far as to use a model, which can be very treacherous sometimes, but not recommended. And then, so what you do is you have your receptor, and you screen your receptor structure here against a whole library of virtual compounds. So these are chemicals, molecules in computerized form. Essentially, they're in a computer. And then you dock them, and then you score them based on you can have like physical chemical parameters, like how well the ligand fits into the binding pocket if it's a receptor. And you can score it from like best to worst score and get some candidate compounds for testing. And another way here is a ligand-based virtual screen. So ligand-based methods here are very different in the sense that you don't use any structural information from the protein whatsoever. But instead of using the structure, you use the actual chemicals themselves. So like the ligands that actually are known to bind and elicit activity with your protein structure, your target protein, essentially, right? So one of the ways to do this is by pharmacophore mapping. And a pharmacophore is essentially, you could think of it as a description of the chemical features of your ligand, right? And this can be anywhere from like hydrophobic regions of your ligand, hydrogen bond donors or acceptors, things such as this. And this is one method that people use sometimes to screen through and try to find similar ligands, chemically similar ligands. Oops. Now another way is by just chemical similarity, which is sort of similar to the previous one, but instead of using pharmacophore, we use other methods to help abstract our ligands, our chemicals, right? And this principle is essentially based on the fact that if you have chemicals, compounds, right, with similar structure, then you're going to have probably a similar function, essentially. So one sort of example of this is morphine, right? So everybody's favorite opioid, right? So this is codeine is also another opioid here, but I mean you can see that they're structurally very similar. They also have similar function. They both bind the opioid receptors. So this is just like one of those assumptions that we're making here for the ligand-based virtual screen. Now, so in order to actually conduct a ligand-based virtual screen, before I go into this here, I want to make it clear that the work today will be presented. It's going to be based on ligand-based virtual screens. So one of the requirements for the ligand-based virtual screens here is that you have to have a set of known data, essentially. So you need some input in order to be able to get some predictions, some output, essentially, right? So one of the challenges here really is in collecting all really relevant data for the method right here, because it's scattered around everywhere on the internet, right? So it can be pretty disordered and structured data. So it's a daunting task. In terms of GPCR databases with pharmacological data that I can use for these screens, there have been databases made already, such as GLIDA, stands for GPCR-Ligand database, and GPCR-Safari. But the bad thing about these databases, I mean, they were good for the time, but no more updates, and we'll see why this is such a big problem. So on the x-axis here is the data sets from like a certain time from one chemical database called Chembl, and Chembl's a database that houses a lot of pharmacological data and whatnot. And this is just you can think of it as like progression in time. On the y-axis here is the number of entries they have, so activities. So like just what kind of experimental values they have, right? So right around this period here is when GLIDA stopped updating their database, somewhere in 2010, and that's the amount of data that we see in Chembl, right? And then right around this time here, GPCR-Safari, they stopped updating everything here. So we have this huge swath of data right here that's just lying around. That's, it's just not really been subset. So, so obviously, I mean, there's a lot of data that's missed right here. So, why not jump on the opportunity to try and subset this here? So, which leads me to the next sort of aim here, which is the construction of GLASS database. So GLASS databases, so the whole objective here is to construct a database essentially to help subset all the GPCR-related pharmacological data to gather all GPCR-related data to gather also ligand-based data, right? And which would be used and also be very useful in computational chemistry or computational biology efforts. Yeah, this is the logo here. I got to say thank you. My wife actually helped out a little bit with the logo design here. I'm not really too good with that. But GLASS stands for GPCR-Ligand Association Database, and a lot of stuff here was done in collaboration with Hongju Zhang, which was not present today, wasn't able to make it for some reason, but very, very good programmer, very, very good guy. So anyways, how is GLASS database constructed? Let's get into it here. So first we start off, the starting point is from UniProt, right? So UniProt, everybody knows, is everybody's favorite protein database, right? So I acquire essentially all the GPCR data for as much as I can from UniProt as a starting point. It's like UniProt IDs, FASTA sequences for the primary structure, gene names, etc., etc. And then the next thing here is I use the information primarily using matching up with the identifiers, protein identifiers, UniProt IDs, to the chemical databases, namely Chembold, BindingDB, IUPAR, Drug Bank, and PDSP. So these are the primary sources of data that I use for the pharmacological data, right? And so I filter these ones for all the GPCR related entries essentially. So I pretty much ask whether or not this data is GPCR related. If yes, passes through. Does the ligand in this entry here of this experimental entry have a chemical identifier? Yes. Pass through, and then also acquire the experimental data associated with this. So it can be anything from KIs, KDs, IC50s, and EC50s, anywhere to just really obscure, just present inhibition and whatnot, depending on the experiment. And so afterwards, I go back into PubChem, and PubChem is essentially like this one of the 14 databases under NCBI that houses a lot of just a really crap ton of chemical information, essentially. And I go back in here and I find essentially match up the ligand data with the ligands from the experiments that were found to be led to GPCRs. So I just get all the information as much as possible. Like chemical identifiers, like SMILES, INCHI IDs are all matched back here. Oxynol water partition coefficient predictions, molecular weights, hydrogen bond donor receptors, and whatnot. All these are all acquired from PubChem right here. And then ultimately, everything's manually verified to make sure there's no mistakes in the data gathering, because sometimes, just a word of warning to y'all, databases can change their format sometimes. And then when you update the database, you lose data. Like, what happened? So manual verification is always important to just check your data, see if it's okay. And then and the way this database is organized here it's just a very simplistic view of how it is. But essentially, we have a table here that's a target. That's the primary key here is linked to an association table. We also have another ligand table here where the primary key is the NG key, which is unique, linked back to the association here. And the association table here consists of all the different associations between your GPCR and your ligand of interest, which is represented by the NG key. And this is associated back to the experimental information, so all experimental data that's found for this essentially. And the current statistics as of Sunday, which is when I updated this year, we have roughly 3,056 GPCRs. 825 of those are human. 687 of those actually have experimental data. In terms of ligands, we have 276,000 roughly, and out of those, 192,000 of those are drug-like, aka they pass the Lipinski's rule of 5 criteria, and for druggability. And in terms of GPCR ligand entries, the non-redundant amount here is 441,667. But overall, we have I flipped it around here. We have roughly 800,000 GPCR ligand entries. Out of those, 440,000 of those are unique. And not surprisingly, analyzing the the distribution of the database here. Most of these are rhodopsin-like GPCRs, the Class A ones. They make up about like over 90% of GPCRs, anyways. So I mean, it's not very surprising. You also have a fair amount of Class C data. Now, Class database essentially you can search by ligand, or you can search by GPCR, or you can even search by GPCR-Ligand pair. So it's kind of hard to show by slides, so I'm just going to have like a really brief tech demo here to show what the database is capable of. So what's everybody's favorite GPCR? Let's just throw it out here. I'm sorry?
CRH.
Oh, CRH, okay. All right, so this one right here. Let's try this one here. Let's try the human one. Okay, so essentially you search for the GPCR, has all the information essentially put into here, and so you have your species, genes, so just like pretty much all the sort of like run-of-the-mill standard information. We also have conditions associated with this that are mined from therapeutic target database. If you go down here for all the human targets, we have a 3D model. If there's no PDB structure available, then there's going to be a GPCR structure model from HGMod, which is a database we have that's pretty much like the predicted structures of all the human GPCRs. And if you go down here, then what's going on here? All right, so you can look for the ligands that bind to it here. So you have like, it's just a list of all the ligands. So if you click on this guy here, you get to go to the ligand page, and this is essentially the ligand page with all the chemical information. And you got a little 2D structure here, and if we go back and you click on the GLASS ID, so this is the association. So you get the experimental information from here. If you go all the way down, then you can see that this guy right here has one experimental value from a study essentially. So this is essentially how this database can be used. You can also download data to play with it if you want also. I don't know why it's moving by itself. All right. So essentially what we have here is we have.
When you in the GPCR page, you have the structure, right? But
Yeah.
do you do domain analysis in this graph with a T4 isosome?
Well, no, we leave that in there. It's just the PDB structure. I mean, we don't process it. It's just for show. So, yeah. Okay. So essentially, the GLASS database here, you can search by GPCR, you can search by ligand. There's also you can search by chemical similarity or substructure if you want also, which I didn't show just now, but you can draw your chemical in and search for it if you want. All the ligands are available in SDF file format. So SDF is like it's just like it's a computerized version of your ligand essentially in 3D format. And they're monthly updates, and the database is fully available to public for download. So you can, do what you want with the data. Now the next thing here is a ligand-based virtual screen. So essentially, I finally have a data set I can use for GPCR screening now. So essentially, the goal of this here is that to perform and validate a ligand-based virtual screen using data from the GLASS database, right? Essentially, why I'm doing this here is because, as I said previously, ligand-based virtual screens can really benefit from like known data, but not just known data. Like the more importantly, we want data that actually it's relevant. Like the ligands are actually bioactive, right? So I have this. So from GLASS here, what I do is I gather all the ligands. So I take all the ligands from GLASS database. So I have all the experimental parameters associated with them, right? Currently, I only work with Ki, Kd, EC50s, and IC50s. So essentially, I have a cutoff value here for at 10 micromolar. I'm sorry? Ki is an inhibition constant, whereas Kd is dissociation constant.
So what is
I'm sorry? Ki is a constant calculated from the IC50. The inhibition at 50%, essentially. The concentrated need to inhibit 50% of your population of GPCRs, right? So it's a constant that's calculated from that, okay? It depends on the experimental conditions. You use the Chang-Prusov equation to calculate back, essentially, right? And so essentially, from this here, I have a pool of bioactive ligands that I sort of like use to filter out here. And all the ligands here are then converted into what's called a molecular descriptor or chemical descriptor. And the one I use here is called the FP2 fingerprint from OpenBabel. So pretty much really generally like how this works is like the algorithm for this here. So this is from OpenBabel for FP2 fingerprint generation. It starts from every atom of your molecule here, it starts from 1 and it goes out to 7. 7 atoms, so it goes like 1, 2, 3, 4, 5, 6, 7, right? And for every one it goes out here, it hashes that feature onto a 1024-bit string, essentially, right? So for one feature, you're going to have like one position on the bit string, right? 1 or 2 or 3, depending on how it's hashed. And then so for all these features here that are hashed, they're all combined together for each feature here into one 1024-bit string. So for each bit here, each bit or combination of bits represents one chemical substructure or like chemical feature. You can think of it that way. Does that make sense to everybody? Hopefully. So it's just a string of zeros and ones essentially, and the ones are the sort of like they denote some sort of a chemical feature. Okay. And this is really more like what the data looks like when I do it. So I start with like the SDF file for the ligand at hand here. And using OpenBabel, I convert it into a hexadecimal string. For some reason, they output hexadecimal, so I have to convert it back into the binary from there. So I end up with a 1024-bit binary string at the very end. So this guy here represents this guy essentially. This does not include stereochemistry. It doesn't include. It's a very. You can think of this as an abstraction of your molecule essentially, right? And the reason why this is used instead of like other methods here is because it's very fast, and I have a lot of compounds to screen, so this really saves a lot of time.
So your phenol and 2-hydroxyphenol has the same fingerprint.
It's probably a little bit different because of the hydroxyl group there. So you're gonna have a different hash that position in the hash that.
Already has the hydroxy group, but if you have one evaluating whether you have one hydroxy group or 2 hydroxy group.
Well, it depends on the position in that case, because it's path based. So you start from like 1, then you move to the next one, you move to the next one. So if you have like OCCO or if it's this way, then OCCCO or something like that, and as opposed to like just one, you're gonna have that additional feature that the other one doesn't have.
You sum them up, right? So
Yeah, you sum them up, but if you have 2 hydroxyl groups, that path, okay, so that path is gonna be, you're gonna have a different path that the other one doesn't have, right? It's gonna be half somewhere else, so it's gonna be, you're gonna have like maybe a couple more bits that are different, right? You're still gonna get some similarity because you still have the aromatic the portion that's similar, but you're gonna get the difference in where the hydroxyl groups are, 1 versus 2, right? Okay. So yeah, so it's represented as 1024-bit binary strings. So the next step here really is more of like the bioinformatics approach combined with the chemoinformatics. So this part here, essentially, I use PSI-BLAST in order to determine functionally similar GPCRs by sequence identity, essentially, right? So PSI-BLAST. And I use all the GPCRs I have from GLASS that were denoted as bioactive, so they had at least one ligand that was bioactive, right? And let's say, for example, we have GPCRs A, B, C, and D here, and let's say after PSI-blasting here, we have B and D are considered homologous. So in this case, hopefully, probably functionally similar, because this really relies on the assumption that similar GPCRs will bind similar ligands, right? And so I do this for every GPCR, and eventually, what happens is I go back to GPCRs B and D, and I take the ligands the bioactive ligands that are associated with B and D, combine and pull them all together into what's called a ligand profile. So what the ligand profile is with my experiment here is the collection of all the ligands from the related GPCRs essentially to your query GPCR. Okay. Now the experimental design here for the validation of the screen is so I start here with the GPCRs with bioactive ligands. So I only test on this subset here because I have the knowns and I want to see if I can use the method to get the knowns back out essentially. And the main thing I want to do here for this experiment is to see whether or not I can only use the ligands from related GPCRs and see if it still performs as well as if I still had the ligands of the query GPCR spiked into ligand profile also, right?
This is for human only?
This is for no, this is for all species essentially. Yeah.
Have you screened out olfactory receptors?
I'm sorry.
Have you screened out olfactory receptors?
There could be some in there. I haven't checked specifically.
That's what the majority of
Yeah.
PCRs are.
Yes, that's true. Like most of the Class A ones are that. I don't know how many of those are in the bioactive ligands here. I could check later on, but the majority probably.
But if that's all
Probably not actually. So yeah, that would be pretty scary if it was humans. So yeah, so pretty much the whole gist of this is here I want to test the ligand profile with and without the ligands from the query GPCR. Okay. And so how does it work? Essentially, what I screen against is a database called Zinc. So Zinc stands for Zinc is not commercial, like the worst acronym in the world. But it's a database developed by I believe housed in Brand Choichet's lab, Choikit over at UCSF. It's a really large database of purchasable compounds. They have like over 35 million compounds. They're all purchasable. They also from like other like vendors and whatnot. So you have like very common compounds. You have boutique compounds. You have just like anything out there you can find, hopefully. And everything is available in an SDF format, like 3D SDF or even like Mol2, so, which is available, which is perfect like for docking. It's all ready. And then so I use the Zinc A database here, and also I spike in the reference ligands for each GPCR that I'm testing against, right? So these are the reference compounds. These are the compounds that are known to be bioactive, essentially, right? And the whole point of what I'm doing here is to try to fish these guys back out to see if I can use my ligand profile to be able to pull these guys back out, right? And I screen this against the ligand profile for the GPCR that I'm working with here. So this is just for one GPCR, right? So so you can think of this as like a pairwise comparison of chemical similarities between all against all. Every single compound in here against every compound in here, essentially. And I calculate the chemical similarity. The chemical similarity is calculated using what's called the Tanimoto coefficient. I don't know if anybody's heard of that, but it's another word for its Jacquard index.
I have a question.
Yes.
What is the I?
Oh, for I and yeah. So
J.
yeah. So in J. So for every single guy here. So every I is every compound in this database. So like 1 to like 6 to 8,000 or something like that. And then for each of these here, I calculate the Tanimoto coefficient for each of these. So J is like each ligand in the profile.
What's the meaning of TC?
TC, I'll get into that. It's Tanimoto coefficient. Okay. Yeah. So the calculation for Tanimoto coefficient. Oh, yeah. So this is how it works. Sorry, I should do that here. It's been a long day. So
TC is based on the structure of ligand. How to evaluate the ligand similarity?
I'm about to talk about that right now. Yeah. So So we do like a bitwise comparison essentially against two molecules. So we have 1024 bit string for one, and we have another 1024 bit string for another molecule. We take those, we compare them against each other to see how many of those ones match up, right? We want to see how similar they are. So the calculation performed here is as such here. You can see all the bits that match up, right? So all the ones that like are union, I guess in this case. So you have like union here, so it's 2, right? And then you divide it by, so 4 is the number of bits in one string, in one molecule, and the other one is 5 is the number of bits in the other molecule. And then you subtract that by the union, or intersection, I guess, in that case. Yeah, sorry. Yeah, I get that switched around usually. Thanks for that. So essentially, yeah, so you divide that by that, and you get 0.29. This is on a scale of 0 to 1. So what you get is, so you can 29.29, you can think of it as they're 29% similar, pretty much, okay? Okay. So for each of these guys here, and so for all of these ligands in the database, so all the compounds in zinc database and all the ligands in the reference compounds, I essentially score them with a series of 13 rules and compare how well they do essentially, and then I rank them. And I have to give a little bit of a word of caution here. The next 13 slides are going to be really, really dry, so it's just describing the rules. So just brace yourself. So the first rule here that we use is called the pre-sum rule, and this is essentially we sum up all the ligands in the ligand profile, like each position for every single ligand. I take the average of like this column, this column, this column, this column, and I get a ligand profile that's sort of like this. The calculation for the Tanimoto coefficient is a little bit different here. It's not I'm not doing bits, but instead I'm doing essentially multiplication of the one times this one or something like that. So I'm essentially multiplying each position of your target ligand molecular fingerprint versus your summed ligand profile. Okay, and the calculation is very similar, not exactly the same, but you guys can sort of get the point from this right here. And so this is one way. One rule that I use here to sort of like help validate. The score is essentially the like what I calculate here from the Tanimoto coefficient, right? And the max rule here is more or less so for every single ligand in the ligand profile, right? So from before I do a pairwise comparison all against all. So this takes the maximum Tanimoto coefficient that I calculate from among your ligands in your ligand profile and use that as the score for your ligand in the zinc database, essentially. So it's just a maximum. The next rule here is the median. So it's more or less along the same lines. Instead of the maximum, I take the median, and I find like what the median TC score is. Sum, it's more or less just the average, add them all together, divided by the number of ligands in the ligand profile, right? And the next rule here is a little bit more interesting. It's essentially the average, but the P here that I divide by is the number of ligands that have a TC score above 0.7, and this is really there to really help sort of like weight the ligand profiles to have more ligands with that are actually more chemically similar, essentially. And the MTA rule is more or less the same. You're multiplying by the sum of all your TC scores here. So the ranking would be inverse essentially for this one here, right? The UC rule, that's just the Euclidean distance. You sum of all the squares of your TC scores, and then you take the square root of that. And the MTA rule is the more than average rule. So essentially what I do here is I take the sum of all the TC scores that are above the mean, and after that I divide it by the number of ligands above the mean. So that's really to help help weight like your ligand profile like higher for if you have more ligands that are chemically similar, essentially. And MTM rule is more the median, so along the same lines of that in the previous rule right there. And so the VAP rule is it's one of the more interesting rules here. So what happens here is you rank all your TC scores that you calculated from your ligand profile, highest to lowest. And the whole point of this rule here is to try to find a gap, like between two, like TC scores. And how we calculate this is whether or not there's like if you times one of those times 1.5, and you try to find to see whether or not it's greater or less than the one that's above it, essentially, right? So let's start here. So 0.8, and then we take 0.76 times 1.5. So that's greater than this here, so there's no gap, right? But if we take 0.76 and then 0.4, so 0.4 times 1.5 here, that's less than 0.76, right? So we consider that there's a gap there, essentially, and we take everything above that and we average it by the number above the gap, essentially, right? And otherwise we just use the average. Okay. And for the NGX rule here, it's very similar to the VAP rule described previously, but instead of the average here, if there's no gap here, then we use the max rule. Okay. And for the NGM rule, more or less the same. If there's no gap, then we use the MTM rule, or the median. And last but not least, just the MTA rule here. So it's sorry about the dryness of the slides here, but anyways, with the so we have all 13 rules here that we use to score like each ligand in the database, see how well it was able to find the true positives. So the way that we evaluate how well the screen actually did here is by enrichment testing. So we do enrichment testing on the top 1% of the compounds. So we rank them all from highest to lowest, or lowest to highest, depending on which rule that I used. And I took that top 1% there and see how many of the reference compounds I was able to get back, essentially, right? So it's calculated essentially the number of true positives here out of the total number of true positives, right? And then times 100. And so you get something between 0 and 100. The 0 being that you didn't find anything, this garbage, and 100 being that you found pretty much everything, right? So the results are as follows. So most of these rules seem to do, it's kind of all over the place, really, but one big highlight here. Well, okay, let me explain what the axes are here first. So, well, okay, so for max rule, the whole point here is it's a positive control, right? So for in the ligand profile, the reference ligands are spiked into the ligand profile, right? And so this one's spiked in. For this one, it's not there. The ligands are not in the ligand profile. Okay, so we're relying completely on the related GPCRs in terms of ligands. Okay. So what we found with the max rule here, 100% obviously for the max rule, because the ligands are they're spiked in the ligand profile, so you're going to pull them out. But after taking those guys out here, you still have you still have like it's decreasing performance, but it's not that much worse. Same kind of goes for the ANZ rule here and the NGX rule, which seems to perform pretty well. All are up somewhat close to 80%, or at least. So the performance remained relatively similar for most of these rules here, which is sort of promising. So essentially, that's really all I have for this part here. So a quick summary of the virtual screen here is that I was able to conduct the virtual screen here, and I'm not sure if you can call it a benchmark, but it's based on ligands from homologous GPCRs, right? Performance is not drastically reduced after you take away all your reference compounds in your ligand profile, and the max rule seems to perform really the best so far. So that's kind of cool. Yes. So last thing here, I'd like to end with some conclusions and final directions. GLASS database is so far we think it's the most comprehensive source of pharmacological data on GPCRs that can be used for computer-aided drug design. Based on the virtual screen benchmark that I had over there just now, it suggests so far that the method could possibly be applied to discover novel compounds, but a lot more work needs to be done right here. So some future directions here. It would be really interesting to see how the effects of the different scoring rules, like what kind of populations of the reference compounds that it captures, right? So max rule may perform the best so far, but other rules may pull ligands that the max rule didn't pull, essentially. So it would be kind of cool to see like how it sort of affects this. So to look at the contributions of each scoring rule, and also maybe try using different combinations to see whether or not it would affect the performance, right? And another future direction here, which I couldn't get in time for this year, which I wish I could, but was incorporating Coach. So Coach is a software that's developed in our lab that's used for predicting binding sites and potential ligands that your query protein may likely bind, right? And I'm using this essentially to try to use a predictive method to incorporate predicted ligands into my ligand profile to see if that increases performance whatsoever. And it would be really cool to see this here because using some kind of a predictive method to increase your effectiveness of your virtual screen would be very useful, I think. So. And most importantly here is the using a larger data set. So the current screen here is done with the Zinc 8 database, which is also the data set they used was sort of like processed. So we used this from like a previous study. That all the ligands were chemically diverse. That they were it was quote unquote a non-redundant data set, right? But more importantly is if we use a larger data set from Zinc 12, 22 million compounds, drug light set, to see if we can get some very similar performance that way, to increase like the breadth. If we get like maybe 10 enrichment factor, then that pretty much means this method doesn't work. So I'm hoping by increasing the scope of this here, it's not only more practical because when you're screening for compounds, you're going to have to screen like millions of compounds anyways. So I might as well try that out and see if I can retain the performance, hopefully. And I think most important of all, like the most applicable thing that this method can do here is for the deorphanization of GPCRs, because there are roughly, I would say, maybe 150 or so GPCRs that are still classified as orphans, Class A ones. So they have no known function, they have no known endogenous ligands, so nothing's really known about these guys. So this could be a very useful method, I believe, for the for trying to help fish out like what kind of ligand it might bind, test out to see if it may elicit any function with the GPCRs, if it binds or if it has any function. It's just stuff like that to help elucidate that. So that would be very nice to see. So I'd like to end this talk with thank you all for listening. I'd like to thank my boss Yang Zhang for his patience and unwavering dedication, as well as Brandon, Jarrett, Kevin, my lab mate, and all the rest of my lab mates here, which I couldn't fit on the page. My dissertation committee, and Beth, and especially Jonathan also, because without Jonathan, our cluster would be like big, huge mess essentially. So. Anyways, thank you all for listening, and I'd love to answer any questions if y'all have any.
When you make a drug profile, how many GPCR homologs do you usually use?
How many do I usually use? So my current cutoff right now for GPs for the PSI-BLAST is 1 times 10 to the negative 100 currently. I'm planning on changing that, cutting it back a little bit, but it's you get anywhere from like 5 to 10 usually. Yeah.
So PSI-BLAST is good for detecting remote homolog in terms of sequence similarity.
Yep.
But it seems that you don't need that much sequence. So why not just try BLAST P, and it will give you less sequence. And
okay.
Yeah, because PSI-BLAST will really give you very remote homolog.
Yeah, that's true here.
Not with that cutoff of 10 to the minus 100.
But
There's just really no reason to use PSI-BLAST under those circumstances.
Yeah. I could just do like a normal blast, I guess, and like the blast P. So yeah, that could be. But you know, good suggestion though. Give it a try.
How many times when you scan the zinc 8 for one query target?
How many times do I scan zinc 8?
Database.
Depends how you sit. Like if I mean I go through every ligand, so once. Because each time I go through the ligand, I'm calculating all the similarities between every ligand in the profile. So you can think of it as I'm going through once.
I mean,
Yeah.
how many times?
How long?
How long? Okay. Let's see. For one GPCR, it probably takes around 5 hours or so.
5 hours.
5-6 hours, yeah. That's for 68,000. So there's the whole computational efficiency problem too. So.
I think maybe you can. One method is to shrink the zinc 8. You can filter out some ligand that is only maybe related to GPCR.
Well,
GPCR related.
Well, the problem with that is that I don't know which ones are GPCR related in the zinc database. They're all unknown, so that's why I want to screen them all. This is the whole point of the screening, because I don't know which one's GPCR related. If I knew, then I'd be able to choose it, but I don't know. So
another method I think is to group the ligand to different groups and their similarity. Then you just can only scan the group, not for every entry of ligand. Because
okay, yeah, that's
groups, you don't care about that. You just only care about with the high similarity ligand group.
right. So you're saying you want me to cluster like ligands, I guess, or something based on chemical similarity. So this is going to be it's possible. I mean, yeah, it might increase the speed. Possibly, yeah.
When you use drug bank, right? Drug bank ATC code.
I'm sorry?
In drug bank, you have ATC code.
Yeah.
So the clinical and chemical feature of a drug assigned by WHO, then that is a measure of chemical similarity.
Mm-hmm.
Perhaps you could try to use that for clustering.
Maybe. I mean, for I have to look into it. I haven't tried that before, but we can talk about it afterwards, probably something like that. Yeah. But it would probably speed up faster if I did that way. But it's kind of hard to say. it might be more generalized in that sense. But I could maybe do that. Look for targets first, like maybe clusters, and then screen those clusters or something like that. That could be really
Really stupid question, but
no stupid questions.
For GPCR structure, how do you know which is the top and which is the bottom?
How do you know which is the top and the bottom? Like what do you mean?
Could you turn to the page of your orphan GPCR?
This one?
How do you know whether the top is extracellular or the bottom is extracellular?
Well, okay. So this is just a crystal structure, but I mean, you see a ligand bound here. You look at a ligand, that's the extracellular right there. And so if you have a ligand bound structure, that's a very obvious. It's going to be right here. If you have like a full length with the, I mean, like they don't have any structures of like the N-terminal domain or anything, but usually, if you have like a large N-terminal domain, it's pretty obvious sometimes because a lot of these guys have large domains that they can't crystallize. They can't have along with the crystallization process. So, but if you model the domain in, then it should be, it's going to be pretty obvious. The size log tail is usually not very large, so that's one way you could tell, I guess. But otherwise, they have 3 loops on inside and outside, so it's kind of hard to see. So, I mean, if you see that, if you look at that disulfide bond, that's on the outside, not on the inside. So that's that way too.
So do any of the orphan GPCRs map close enough to any of these things that you can start making any predictions?
Yeah, there are some that I've checked already. So far, because my cutoff for my e-values it's pretty stringent right now, so it's kind of limited in scope. So I'm actually trying to tune my e-value right now, like a little bit more flexible. I actually had a committee meeting on Monday. We talked about this problem. One way I'm thinking right now is to reduce my problem to the Class A GPCRs and try to look at only homology with the transmembrane domains instead. And from there I could probably loosen my e-value cutoff to try to find like close but not like too far away homologs and stuff like that.
And that's what
So,
we're all hitting on in any case.
yeah,
Just the transmembrane.
exactly.
I don't think isolating them will make any difference for you. Problem.
Yeah, yeah. I mean, one thing I'm kind of concerned about is like whether or not, because like there's some with like these really large, like N-terminal like domains, like the adhesion proteins and everything. So yeah, true. Yeah, like the Venus flytrap ones, right? The Class C ones or something.
I forget which.
The methodic limit. Yeah, I think it's Class C. Yeah.
Calcium receptor like that.
Yeah. Yeah, those are they all usually have like pretty big ones, so that might influence the e value of the thing. So I might look at only the transmembranes for only Class A's, anyways. Yeah. Yeah.
What the ranges are on those scores, there's the transmembrane regions are still going to dominate.
Sure.
You might, because you've increased the length by 2 perhaps, for some of the really huge ones, it may cut the e-value as much as 2. You have to play.
Yeah. Probably play around and see like, how much it affects. Yeah.
It's not that big a difference.
Yep.
How many open GPCR?
About like for humans, about 150 or so for humans, according to IFR classification. Yeah. That they're mostly Class A, most of them, which is not surprising because they're the largest group anyways. So. Yeah.
Thank you.
All right. Thank you.